Hi, friends. Welcome again to our series of Histology made easy with diagrams and drawings. Today we're going to talk about mitosis and meiosis, things that you have probably heard many times. Our purpose today is to try to understand more and not to memorize and say just simple regurgitate, saying like prophase, metaphase, anaphase, telophase. But trying to go a little bit into understanding, and for that we also have to mention the cell cycle, which is pretty much the history of a cell, pretty much the life history of a cell. Let's try to explain what I mean. For example, if we have a cell that is going to be pinched in the central portion and divide into two cells, right? This is the process of for the most part, mitosis. What happens to the cells next? Well, that is the story of most cells of our body. To begin with, just days after conception, the embryo starts making decisions into what they are going to be. For example, some cells are going to differentiate into a placenta, and others into the embryo itself. So that is a big decision at the beginning, just 5 days after conception. We're talking now of a process that we would call differentiation. What does it mean? It means that the cells are going to look different and function differently than the others, although they all come from a single cell, from a unique cell at the moment of conception. But they start differing from each other. Some of them will end up being very long, or it's very short, or small, or the different sizes. This is the process of differentiation, which we'll have to also remember. In addition to that, the cells have to just do their function. In other words, they enter into what they called growth phase 1, or G1. What do they do in this phase? Well, if you look at the microscope, just about any organ, the cells are going to be doing their business. Whatever they are not multiplying like rabbits. They are working, and they need to work to make our body function. So in other words, the G1 phase, the cells are, let's say, for example, simply working. If you look at the microscope, you're going to find out that occasional here and there, you're going to see mitotic figures, mostly in metaphase. They're easy to see as a dark spot. But for the most part, they don't divide, and they can be like that for, let's say, for days or even months, depending on the tissue. But they will reach a point in which the cell has to divide in many tissues. So when the cell divides, she will reach to this point of mitosis. But we have to be careful what we mean by cell division or replication. In other words, for example, if we use our naive idea that a cell just simply pinches itself and divides, that doesn't really work. For example, let me see what I mean. For example, if you have a cell with a nucleus that's very dark, usually, and we imagine the cell dividing simply by pinching, let's say, in the center and dividing in two cells, that would seem pretty logical, right? But it's not quite logical because the two cells would only have half of the chromosomes. In other words, the two daughter cells would inherit half of the richness of the first cell. That's not very kosher. So in other words, in order to have to divide, to have a mitosis, there are two conditions that we have to reach. But before that, let me just mention a little bit what a cell is really. If you remember, a cell that's not dividing will be, just a second, excuse me. It will be looking pretty much like this. It has very dense chromatin, mostly at the periphery. It has heterochromatin and euchromatin. And well, this is, what does it mean? In a cell that's dividing, the genetic material is all loose, let's say. All the chromosomes are loose. Now, how are you going to divide a cell that has the chromosomes loose like that? You need to do something special. I wouldn't worry too much about pinching the cytoplasm because the organelles can be replaced later. But the nucleus is a big problem. I forgot to draw here the nucleus. Okay, so what is really going on in this cell if you focus on the DNA, on the chromosomes? Well, what you would really have is something like this. You would have a nucleus. If you could really look into this cell, you would see that these chromosomes are going to be like this. That would be, for example, one chromosome, a paternal chromosome, and for example, this would be a maternal chromosome. Now, you tell me, and we have 23 pairs of those chromosomes. Now, you tell me, how are we going to divide the cell that has all these chromosomes loose inside this cell with that looks almost like a geographic appearance of a cell? Well, yeah, we have to have two conditions in order to divide a cell. First, the genetic material has to be duplicated in order for each daughter cell to get exactly the same amount. That's actually common sense, right? And second, the genetic material that is totally loose has to be packed, has to be really, I would say, just made into packages that are easily sorted to one cell or the other. And those conditions are met in the mitosis, those two conditions. The daughter cells have identical amount of genetic material, and that genetic material has been duplicated like that. Now, how do chromosomes look like? I mean, let's just remind ourselves what do chromosomes look like. I have too many markers here. Okay, one chromosome, if you met in an interface like this, would look like this. Just a long strip of DNA and histones, etc., etc. But this would be the paternally derived chromosome. We'll put it blue because it looks more for a paternal chromosome. And the maternal chromosome would be like that. In other words, we know that chromosomes come in pairs, each one coming from the father and the other from the mother. So we have 23 pairs in a total of 46 chromosomes. We can actually start drawing and drawing. I could perhaps, just for the sake of example, start to showing you how would another pair would look like, exactly like that. Now, in order to understand how we need to duplicate these chromosomes. In other words, we need to have a situation in which this paternal chromosome will be duplicated, it was replicated, and this maternal chromosome will be duplicated. But even then, we would have a problem. If in the nucleus we have a situation in which the maternal and paternal chromosomes are duplicated, there would be still in a loose situation. We need to package them to make them short. Before doing that, I'll remind you a little bit about whatever is going on with the DNA. Of course, you guys are young and know well about the DNA. I don't need to say anything about it, but let's, for the sake of completion, talk about the DNA a little bit. The DNA is a molecule that goes like this, and everybody knows, those who are not in the biological, they know that the DNA looks like this. Okay, we know that they actually, we have here sugars that are, but let's actually make it like that. If we untwist this helix, we have a ladder kind of situation in which we have the, let me see, we have the deoxyribose here. I want to erase the whole thing. I'm going to make a ladder make of deoxyribose. Like that. Bear with me, be patient, because this is for you, for your benefit. Okay, if we have another chain made of deoxyribose, which is a sugar, which looks pretty much like this, like a pentagon. We know that these deoxyribose are joined together by a phosphate moiety, like here. And here, pretty much like this. So we are getting pretty close to what DNA is. And we need actually something that is very important, or the nitrogenous bases that are in the center. And you know that those only 4 nitrogenous bases that are very, go directly, adenine, thymine, cytosine, guanine, and et cetera, thymine, adenine, et cetera. So you know that this is pretty much the essential, the essence of what DNA is, which was discovered about pretty much 70 years ago by Watson and Crick. And we found out that 3 of these letters are a codon, which are referred to a single amino acid. So that means what this information is copied by the messenger DNA. The transfer DNA will be able to understand and bring amino acids and form the protein. But this is it. Now, if you remember that also RNA is not very different from DNA. It actually thymine is replaced by uracil, and deoxyribose is replaced by ribose. But also we have a host of other proteins which are histones around it. Now, in order to, we're trying to understand pretty much how DNA duplicates. It is not that, it's in principle is not that difficult. This chain will be like that, will be opened, and another chain will be formed. There is an enzyme DNA helicase that separates those strands, and there is a DNA polymerase that will populate those new nitrogenous bases in going in one direction and in opposite direction. One going in this direction and the other one going in the opposite direction. Well, now it gives you an idea of how DNA can be replicated, and whoop, it looks like it's going to look, instead of one single strand, you see two strands because of these enzymes and this kind of simple mechanism, I would say, of duplication of the DNA. By the way, this happens not only in us, it happens in all animals, in all plants, and even insects, all eukaryotic cells. So let's go back a little bit to what we were saying. Now, we know that the DNA has been duplicated. That's the first condition. The second one, it has to be packaged in small packages, I would say. And for example, we would have here, let me get, if I have too many markers in my hand, if you have the male chromosome will coil like this. And they are joined by the centromere. Obviously, it does look duplicated. It does, doesn't it? This is much shorter than it was before. And the female, actually the maternally derived chromosome, I should say, is also very, very, very much coiled. So the two conditions for the mitosis are now here. We have a duplication of the DNA, and we have a packaging of the genetic material so that it will be sorted out well between the two daughter cells. Now, the process of cell division goes as follows. At the beginning, we have a first phase, which we call the S phase, which is the synthesis of DNA. It's called the S phase. And then before going into the division itself, the cell has to check everything, that everything is correct, including the coiling of the chromosomes, in another phase, which is the growth 2 phase. It's like a little phase in between the S phase and the M cells for mitosis itself. So in other words, this is what we have: the cell cycle, the cell doing its business most of the time, and once in a while dividing into two daughter cells. This is a process that looks very simple, but we are made of trillions, 40 trillions of cells. That means many, many, many divisions, and that is it. Now, before I forget about something important, some cells just once they are formed do not divide. Growth 0, they don't multiply. And those are, for example, cells like the neurons, like in our brains, that they just, once they are there, they are there. We don't have more neurons. That's too bad. And however, just to make our life more interesting, we have some cells that don't divide, but occasionally may divide. And that's, for example, the case of the liver cells, the hepatocytes that may go all the way and go back to the S phase and stop in the S phase without dividing. What does it mean? That if you look at the liver, and the liver sees a lot of cells that are binucleated, that have big cells, and are found to have twice the amount of DNA. And there's nothing wrong with that. The patient doesn't have cancer, and that's essentially it. Now, these are facultative dividers. Essentially, that is it. Now, we are going to talk about mitosis itself. Mitosis is we usually tend to jump from prophase, metaphase, anaphase, telophase. Actually, this is an evolving process, and it's easier understood, as a slow process, not as just something that you have to memorize. Let's say, for example, when we start in the mitosis, and that's what we're going to do right now, we have a cell that has at the beginning, we're talking about at the end of the G2 phase, essentially, and we have a good amount of chromosomes that look like this. Those are the paternally derived. I'm only painting 2 instead of painting all of them. This will be, for example, the maternally derived chromosomes. Okay, and of course that would happen to all the chromosomes. I'm just putting here 2 pairs. So you excuse me not to draw here 32 pairs because I don't even have room for that. Now, there is one important component that will actually guide the cell division, are the centrioles. The centrioles are divided usually in the S phase into 2 different centrioles, which are joined by microtubules. What we see here is a cell that that has its chromosomes already visible but clumped all together in the nucleus, and this is the prophase. Okay, now what is going to happen next? Next is going to happen that I get rid of this text, a little bit difficult to erase completely. The centrioles, which they were moving to the opposite sides and are joined by microtubules, are going to be useful, are going to be as a framework for all the chromosomes to be attached here. So for example, I'm going to just draw 1 pair, and that will be, for example, the paternally derived chromosome and the maternally derived chromosome, the maternal derived chromosome. You see that those are 2 copies of the same, 1 copy of the same chromosome. I will say they have 2 copies. They are joined by the centrosome, and you have now we are dealing in a situation in which this is called metaphase. In the metaphase, we have almost a dark band of chromosomes right in the equatorial portion of the mitotic spindle. And you know that the genetic material has been multiplied, it is packaged, and now it's ready to be separated, which happens. I hope I can erase this because it doesn't erase too well. One copy of the paternally derived chromosome will go to one of the daughter cells, and the other part is going to the other, and the same thing is going to happen to the other chromosome, maternally derived. Essentially, we're having a total democratic division in two cells, in which both cells have identical amount of DNA. Now, we're not dealing now when they appear to start to separate, we just don't call it metaphase, but we call it anaphase. So when the chromatids separate, we have the anaphase. The anaphase travel along the microtubules to the vicinity of the centrioles, and pretty much at this point, we are reaching the end of the mitosis. And sorry about these markers. Let's use the other markers. We have now the paternally derived chromosomes one side, and maternally derived chromosomes to the other side. But this is happening to 32, to 32 pairs of chromosomes. So this is much more dense, it would say, but essentially is the same. Now, what is going to happen? The cell has already divided the genetic material in both sides, and the cell starts to undergo cytokinesis, which is the nucleus. The cell starts to divide, and the nucleus gets formed on each cell, and we're going to have this stage of the telophase, which is the final stage, telophase, we have two daughter cells. Now, the cells are going to look a little bit smaller than the original, but as soon as they begin to work, they can modify the amount of organelles, and they're going to look exactly like any other one. They're identical cells. Now, we're going to discuss the issue of meiosis, if you bear with me. Now, meiosis is an interesting situation. We have to produce cells that have half the amount of chromosomes in order for that cell to join another cell from another person and form a different organism. So in other words, we need to divide to get cells with half the amount. Now, for that, we have only two organs who do that kind of operation, I would say, which is the testis and the ovary. The reproductive organs are made essentially to maintain the species for that purpose. Now, in order to we have actually, there are two meiotic divisions. And luckily for all of us, the first meiotic division is essentially the same. If we, for example, recall that in the mitosis, you have the centrioles, and after the prophase, in the metaphase, we have the paternally derived chromosome and the maternally derived chromosome, and they divide. They separate and go to different places. Now, the very, very, very important thing to remember is that in the first meiotic division, this happens, but it happens with a very important manner, I would say. The paternal and maternal derived chromosomes don't just divide with two chromatids going in opposite directions. They actually, if this is the paternally derived, they get very much close and personal with the maternally derived chromosome. That's, in other words, they get very, very close, just as close as the father and the mother got before. And that has a very, very important reason. It's to produce cells that are going to be different. There's going to be genetic diversity. In other words, here is going to be an exchange of genetic material between these chromosomes, from the paternal to the maternal and vice versa, at different places. So at the end of the first meiotic division, you're going to have, in the anaphase of the first meiotic division, you have a paternal chromosomes going in different directions. Let's say maternal chromosomes going in the opposite direction. And however, there is a big difference from mitosis. The paternal chromosome is going to have regions that correspond to the maternal chromosome, and the maternal chromosome is going to have regions that correspond to the paternal chromosome. So that makes it a variation. After that, the cell divides, and if you will bear with me, we have two cells. We will have two cells, and we'll have two cells in which the paternal chromosome will be here, and the maternal chromosome will be here, with the difference that the paternal chromosome is it is not the same. It has received exchange of genetic material, and this one too. Now, this is the first division of meiosis. The second division, these cells, they just undergo division without S phase. In other words, the second meiotic division is the only division that takes place without DNA synthesis. In other words, we're going to get this situation like this. Finally, this cell is going to divide into those two cells, and this one into two cells. And for example, these are going to be having one copy, and then this one are going to have this one copy here, and this one only one copy here, and this one only one copy here. In other words, they will only have one copy. They only have 23 chromosomes, not 46. Now, we know that these chromosomes are different. In other words, we'll go to that later. Now, what happens in the male? In the male, obviously, the cells mature into spermatids and then into spermatozoa. Spermatozoa, because that they have a long tail, because the cell already knows that it's going to have to go a great distance, almost 12 centimeters, to reach the ovocyte. And in case of the female, these three of these usually die or just degenerate, and only one of them remains. Now, what is the difference now between the male and the female? Now, in the male, we're pretty much behind, I will say, the male. The spermatogenesis only starts when the person is 12 to 14 years old. In the meantime, we only have just cells that have not started meiosis, like spermatogonia. They're pretty behind. How about women? Women go far, in front of us. In women, the mitosis starts when the embryo, in the fetus. By the 5th month, already the female may have up to 8 million ovocytes that are stuck in the prophase of the first mitotic division. Already, it's as though the little female the female body would understand that one of its functions, more important, is to provide cells for reproduction. From those 8 million cells, usually about 1 to 2 million are present at a time of birth, because the other ones degenerate. And then forward, every month after 12 years old, after the menses, a cluster of cells will go, but only one of them will be ovulated. That is essentially what happens in meiosis. So, I think that we're pretty much covered most of this material, and I hope it is useful to you. Have a good time.